In Chapter 15, we have two major goals. Goal number 1, we do want to take a look at glycogen metabolism, both the production and degradation. And our other major goal is to really understand how the overall glucose metabolism is regulated. This means we need to understand how both glycolysis and gluconeogenesis are regulated, but also consider how glycogen metabolism is regulated. So the first thing we can do is recall the important steps of glycolysis and the reverse gluconeogenesis. So with glycolysis, you see these three reactions where the enzyme is noted in red. Those are going to be the major reactions that we will regulate. Conversely, as we look at gluconeogenesis, it's the corresponding reactions that we will note are highly regulated. If you'll recall from the last chapter, these regulated sites are the places where typically energy is going to go into the reaction or be produced in the reactions. So they serve as very good control points. So as we start jumping into this regulation, we want to first think about which enzymes are going to most affect the pathway. And it turns out, if we take a look at this graph, that it's actually hexokinase that affects the flux or the movement through glycolysis more than even phosphofructokinase 1, which is a highly regulated enzyme. So let's think about how is hexokinase regulated. One way that we regulate hexokinase in our body is specific to different tissue types. In different tissues, we have different forms of hexokinase. And so two that we can compare very easily would be glucokinase, shown here in red, that's found in the liver, versus hexokinase 1, which is found in the muscle, brain, and other areas like that. So if we think about and look at these graphs, we can notice that the Km value for glucokinase is much higher, a much higher glucose concentration than the Km value for hexokinase 1. Now what this tells us is it takes a much higher concentration of glucose to reach the maximum velocity ability of glucokinase. In the liver is much more concerned with sending glucose out to other tissues, so glucokinase only functions maximally if we have a lot of glucose around. If we compare this to the muscle that has hexokinase 1, it works at even very low glucose concentrations, hence the low Km. The muscle doesn't care about the other areas' ability to use glucose. Hexokinase activity is also regulated by the type of glucose transporter found in the tissue. So here in the liver, we have a GLUT2 transporter that has a very high KT value, transport affinity, which gives it a very low affinity, meaning that this GLUT2 transporter can move glucose in and out at pretty much any glucose concentration. Okay? The high Km allows this hexokinase to work even at those high concentrations. Another way we regulate this enzyme is by a regulatory protein. When we have a large amount of fructose 6-phosphate, meaning we do not need to do glycolysis anymore, we will actually remove the hexokinase out of the cytosol and hide it in the nucleus bound to a regulatory protein. If we have a large amount of glucose brought in by this glucose transporter, that glucose will activate this hexokinase to release the regulatory protein, move into the cytosol, and begin converting glucose to glucose 6-phosphate. Now looking at regulation of PFK1. So PFK1 is activated by AMP and ADP. AMP and ADP tell the cell that glycolysis is needed, so it activates the flow through glycolysis. ATP is the opposite. It tells the cell that we do not need to produce ATP, so the presence of ATP will slow glycolysis. Citrate also slows the activity of PFK1 because citrate signals that we have had a pretty good supply of glycolysis occurring, and we've even activated the TCA cycle enough that we've activated glycolysis. So that citrate serves as a signal that the downstream pathways are going good, so we can slow glycolysis. So now let's look at how this fructose 2,6-bisphosphate allosterically regulates PFK1. So this is the normal pathway where fructose 6-phosphate is converted into fructose 1,6-bisphosphate in the process of glycolysis. Same regulation shown here. The reverse is the pathway for gluconeogenesis. If we have a large amount of fructose 6-phosphate, we'll not only produce fructose 1,6-bisphosphate, but the high concentration of fructose 6-phosphate will stimulate PFK2 to produce fructose 2,6-bisphosphate. So large amounts of fructose 2,6-bisphosphate tell us that fructose 6-phosphate is highly concentrated, so we need to activate glycolysis. So if we go back here, notice fructose 2,6-bisphosphate activates PFK1 because it's a signal that a lot of substrate is available. So what is the regulator for this PFK2 activity? PFK2 is active when it is not phosphorylated. In the presence of insulin, we remove the phosphate from PFK2 and it's in the active form. Therefore, insulin triggers production of fructose 2,6-bisphosphate. So insulin is stimulating glycolysis. The opposite would be true with glucagon. Glucagon activates a G protein coupled receptor, leading to a high concentration of cyclic AMP. Glucagon is going to lead to phosphorylation of PFK2, making it inactive. So glucagon signals there's not enough fructose 6-phosphate to really push through glycolysis. So glucagon is going to inhibit glycolysis, but it will stimulate gluconeogenesis. This is a good point for me to point out to you a couple of things. Insulin signals I have a lot of glucose, use it. So insulin signals go glycolysis. Glucagon signals I don't have much sugar. So glucagon says stop glycolysis. Anytime an enzyme is active in the presence of insulin, that enzyme is active when not phosphorylated. Anytime an enzyme is active in the presence of glucagon, that enzyme is active when phosphorylated. Always true. Then our last enzyme we take a look at is pyruvate kinase. Pyruvate kinase is an enzyme that is inhibited when it's phosphorylated. So pyruvate kinase is not going to function whenever we have the signal of glucagon. And that's what this figure shows you here. In the presence of glucagon, we phosphorylate pyruvate kinase. It is not active. As far as regulation of the pentose phosphate pathway, we simply regulate the pentose phosphate pathway by looking at how much NADPH is available. large amounts of NADPH signal that we should slow down pentose phosphate pathway, and that's what's shown here. So considering everything that we've looked at so far, we understand that the two major fates for our pyruvate is to either serve as a source of gluconeogenesis, or it can continue into the citric acid cycle to produce energy. Insulin is going to trigger going down this red pathway. Glucagon is going to signal going up this blue pathway. Now let's think about glycogen metabolism. Glycogen is your body's way of storing excess glucose. Your liver stores excess glucose so that it can supply the entire body with glucose during low blood glucose times. Muscles store glucose in the form of glycogen so that the muscle itself always has an available source of glucose to produce energy. So this figure serves as a good overview of glycogen metabolism. To break down glycogen, we need an enzyme known as glycogen phosphorylase. Glycogen phosphorylase is activated by glucagon. Glycogen phosphorylase breaks off one glucose monomer from glycogen at a time in the form of glucose 1-phosphate. We then use phosphoglucomutase enzyme to convert glucose 1-phosphate to glucose 6-phosphate. And then glucose 6-phosphate in the liver can be converted back to glucose using glucose 6-phosphatase. This was an enzyme from gluconeogenesis. To produce glycogen, glucose is converted to glucose 6-phosphate, then by phosphoglucomutase to glucose 1-phosphate. Glucose 1-phosphate is converted to an intermediate known as UDP-glucose, which can be acted upon by glycogen synthase to produce glycogen. Glycogen synthase is activated by insulin. So let's look at each of these in a little bit more detail. So how does glycogen phosphorylase work? So shown here, we have a portion of a glycogen chain. One glucose monomer at a time is removed from the non-reducing end, releasing glucose 1-phosphate. This will continue down the glycogen chain until a branch point is approached. When we are approximately 3 glucose monomers from a branch, the debranching enzyme will remove these 3 monomers and place it on the non-reducing end. Now glycogen phosphorylase can continue. The one glucose monomer that's still attached by an alpha-1,6 linkage is released as free glucose. The glucose 1-phosphate that is released is acted upon by phosphoglucomutase. A serine residue found in the active site of the enzyme is going to temporarily house a phosphate. This phosphate will be added to the correct number 6 carbon of glucose, and then this number 1 carbon phosphate that's found on the glucose 1-phosphate is transferred back to the enzyme. So what we end up with is regenerating the correct form of the phosphoglucomutase enzyme and releasing glucose 6-phosphate. One way that we can start thinking about control of this production and some extra things that occur specifically in the liver, whenever the glucose 6-phosphate is released from the glycogen breakdown, this glucose 6-phosphatase will remove a phosphate, releasing glucose. Glucose is then transported out that GLUT2 transporter into the bloodstream. So this is something that only happens inside of the liver. That will be important when we get to regulation. The overall control of this glycogen phosphorylase comes from our two enzymes, glucagon and epinephrine. Glucagon phosphorylates glycogen phosphorylase. If we note what happens in this figure, in the presence of glucagon, we see this phosphorylation of our glycogen phosphorylase enzyme, A for active. Also note that other things can lead to phosphorylation, making glycogen breakdown occur, and these would be epinephrine, calcium, and high AMP concentration. All of these would signal that the muscle needs a readily available glucose source, so that says we need to break down the glycogen. Remember this picture from our chapter 12 when we were looking at signaling? So both glucagon and epinephrine both will bind these G protein coupled receptors, leading to this cascade of phosphorylation, where we activate glycogen phosphorylase by phosphorylating the enzyme, breaking down glycogen, releasing glucose 1-phosphate, convert it back to glucose 6-phosphate, back to glucose in the liver, or releasing that glucose 6-phosphate and it goes directly into the muscle for glycolysis and production of ATP in the muscle. Glycogen synthesis involves this important activated intermediate UDP-glucose. So how do we get UDP-glucose? To produce UDP-glucose, we need a nucleotide triphosphate, and we're going to combine this with a phosphorylated sugar, and we end up with a sugar connected through two phosphates to a nucleoside. So we get a nucleoside diphosphate sugar. This reaction is driven by the release of pyrophosphate. So see, we have 1, 2, 3, 4 phosphates, 2 remain, 2 are released, and the subsequent breakdown of this pyrophosphate is going to really drive this reaction forward. So once we have our UDP glucose, it is going to serve as our single glucose monomer that we can add one at a time to build our growing glycogen chain. The start of the glycogen chain is actually not a sugar. It's a small peptide known as glycogenin with an active tyrosine residue that serves as the initial hydroxyl area for the first UDP glucose to bind. So you can see the UDP is released out and that single glucose monomer binds to glycogenin. This is going to continue until we get about 20 to 30 glucose monomers out from glycogenin, and then the glycogen synthase can actually remove that glycogenin core and keep going. We have a branching enzyme that adds those alpha-1,6 linkages. So how do we control glycogen synthesis? Glycogen synthesis is going to be activated by insulin. Notice glycogen synthase is active when it is not phosphorylated. So in the presence of insulin, we receive the signal that we have enough glucose. Let's store it. So insulin leads to the dephosphorylation of our enzyme. Glycogen synthase is active, and we store the excess glucose in the form of glycogen. Glucagon and epinephrine have the opposite effect because glucagon and epinephrine will make this enzyme phosphorylated. High amounts of glucose and high amounts of glucose 6-phosphate are going to increase the effect of insulin, leading to more glycogen production. Now these last few slides really are just summary slides to put everything together for you. So what's shown in red here is what happens in the liver when you have high glucose. So high glucose triggers release of insulin. Insulin leads to activation of glycogen synthase. So we increase the production of glycogen. High blood glucose also activates glucose 2 transporters to pull in the glucose, and we see an increase in glycolysis. The counter is true in the liver when we see low blood glucose. That triggers the release of glucagon. Glucagon leads to this signaling cascade that ultimately phosphorylates glycogen synthase, decreasing glycogen synthesis. We phosphorylate our glycogen phosphorylase, activating glycogen breakdown. And then we also will lead to phosphorylation of pyruvate kinase, decreasing glycolysis. So let's compare that to what goes on in the muscle. So the muscle doesn't respond to glucagon, but the muscle does respond to epinephrine. So we'll see in the liver, in the presence of glucagon and epinephrine, increased breakdown of glycogen. Remember, both of these signal I need glucose. So the liver breaks down glycogen, and the muscle breaks down glycogen. The liver is going to slow glycolysis, but increase gluconeogenesis. The liver is the only place where we can do this gluconeogenesis. In the muscle, in response to low blood glucose, we actually keep our glycolysis upregulated. The muscle can't worry about what's going on everywhere else. The muscle just needs to make sure it keeps an energy supply for muscle contraction. And that's really what this last figure is showing you here.